All right, to meet my expectations today, you're going to have out on your desk your homework that was given to you on Friday and a grading pen or pencil. So pause the video right now and have everyone get their things together and get ready. All right, so let's go ahead and start grading your homework. We're going to grade this section first. This is referring back to, I don't know how many of you remember that PowerPoint where I had the ugly ice skater we all said looked like Jim Carrey. This goes back to that lesson where we're looking at how living things are organized from the most simple and smallest unit all the way up to the most complex and largest. So it starts from small to large. And so a group of similar cells, or a cell, then makes up tissue. These tissues, when put together, make up an organ. When you have multiple organs working together, they make up an organ system, and all of these things work together to make up a full organism. And that was the Jim Carrey ice skater thing. Go ahead and pause if you need to. Each one of these is worth a separate point. You don't have to have the word organism on there. All right, this next section, the direction said from the following terms, choose the correct answer to each riddle. Now, here's the thing. I had some emails this weekend that said, hey, did we talk about these different kinds of cells? No, we did not specifically talk about these. I was looking to see if you could use some deductive reasoning. This was super easy, and you could use your context clues to figure out what answer should be here. So the first one says, I'm flat and I get tougher as I get older. If you look at your other options, the only option that works is skin cells. Your blood cells aren't going to get tougher. Your nerve cells aren't going to get tougher, and neither will your muscle cells. Okay. It also says to form a protective layer around your body. Nothing else would work for that. Number 2, I'm a small disc and disc-shaped. My smooth round edges help me move easily through the smallest blood vessels in your body. That should have told you right then it can't be anything else but a blood cell, because nothing else goes through your blood vessels. It then continues to say, I deliver oxygen to different body parts and remove waste. So that was a pretty obvious one too. Number 3, I am long and shaped like a wire. And so that one right there, if you stopped right there, you might have been confused. However, when you continue to read the rest of the definition, it says, I can deliver messages from one part of your body to another one in a very short time. Delivering messages is always going to be those nerve cells. Okay, which leaves muscle cell, but let's look at the definition. I am long and thin and can contract, become shorter. And you use, and when it says I use a large amount of energy, that should tell you straight away that it's a muscle cell. Go ahead and mark the ones wrong that you need to mark wrong, and leave the ones that are correct alone. All right, this part of the assignment said write at least one feature that helps each type of plant cell do its job. And I'm not going to lie to you, this might have been a little difficult for you because you really had to sit and think about what you knew about plant cells. But it wasn't beyond what you were able to do. The first one, a leaf cell. Well, you know that leaves are responsible for photosynthesis, and so those cells have chloroplasts in them for that purpose. Number 6, the stem cell. This one you had to really think about, and as a result, I'm not going to have you mark this one wrong if you didn't get it correct. Just leave it alone. But these cells have cell walls, and they are used for rigid support. That's why you can have a sunflower that's 6 feet tall, because the cells in their stems have those super rigid cell walls. Number 7, the root cell. You had to ask yourself, what does the roots of a plant do? Well, they're responsible for sucking up water out of the ground. And so we know that they have vacuoles, and they have really large vacuoles that hold water. All right, let's flip your paper over and let's grade the other side. This one you're supposed to match the job to the organelle, and all of these are things that you've studied and you should know. The first one, those storage areas for food, water, and minerals, are obviously F, the vacuoles. Number 2, photosynthesis takes place in these structures. We just talked about that. That's H, the chloroplasts. Number 3, a gel-like substance in which a cell activity takes place. That jelly is going to be the cytoplasm. Number 4, a flexible layer that forms the boundary around a cell. Also, they could have put in that it's semi-permeable, and that would be the cell membrane. Number 5, these control the activities of the cell. The control center is C, the nucleus. Number 6, these release energy that the cells need, and the powerhouse of the cell is the mitochondria, E. Number 7, this is going to contain the DNA, which is an organism's recipe for all of their traits, and that's going to be D, chromatin, or chromosomes. Either one, I would take either one of those. All right, and number 8, this supports and provides structure for the plant. We just talked about that, and that's G, the cell wall. All right, the last one, or last section of your worksheet for us to grade. This line, number 9, is pointing to that very outside of this plant cell, and that is the cell wall. The lining just inside of the cell wall, it's going to be the cell membrane. Number 11, it's pointing to all of that stuff that's inside of the nucleus. That's going to be chromatin or chromosomes. I'll take either one. Number 12 is pointing to the outside of the nucleus. That would be the nuclear membrane or nuclear envelope. I'll take either one. Number 13 is pointing to that jelly substance. That's your cytoplasm. 14 and 16 are hard. Because the way they drew 14 doesn't look anything like what it should be, and that's a chloroplast. It doesn't look anything like it. So I will take chloroplast or mitochondria on number 14, and then likewise on 16, I'll take chloroplast or mitochondria, just because they were drawn differently than what we're used to. But number 15 is obviously that vacuole, and then again 16 is the mitochondria or the chloroplast. I'll take either one. And the last one, number 17, which cell parts are found in plant cells but not in animal cells? That's your chloroplast and the cell wall. All right, go ahead and clear your desk of everything but a pencil. Have your homework assignment ready to be collected. And today I'm going to provide your note page for you. So that's why you need nothing else but a pen or a pencil on your desk. And for those of you that like to highlight your vocabulary, go ahead and get your highlighter out. Go ahead and pause this video until everyone has all of their supplies and they're ready to get started on notes for the day. All right, so, I've got a question for you. How long do cells live? You think about that. I guess it depends on the cell and how much wear and tear it gets. Like, for example, your red blood cells have an average lifespan of 120 days. So the average red blood cell lives for about 4 months. Your rapidly dying cells, like your skin and hair, and even in your stomach and intestines, these die even faster than 4 months. So that may leave a question in your mind. Wait a second. If cells have an average lifespan, we'll just take the average of 4 months. How come you're still alive? I mean, you should not have even made it past the baby stage. You should have died. And so how do we answer that question? How in the world can you still be living if that's the case? How do you not run out of blood? The answer is in mitosis, and that's called cellular reproduction. Now, as we go through this lesson, I'm going to guide you in your note taking, so don't write anything down until I explicitly tell you to. That way you'll get everything down in your notes that you need to have, and you won't be stressed out trying to write a bunch of things that you don't need to have, okay? So mitosis. Obviously, your cells are going to have to reproduce or you wouldn't have made it past your infant stage, okay? So the first part in your cell notes, if you'll look, I've got an example here of what mine looked like. You're going to want to write cell division. It's a process by which a cell divides into two new cells. Okay, the next thing you're going to write, and I wrote a star next to this because this is a very important question. Why do cells need to divide? Well, things grow by producing more cells. Cells don't get bigger, they increase in number. It also has to repair damaged cell and tissue. And if the cell gets too big, it will then lysis or die. Go ahead and pause the video right here and get these notes down at the very beginning of your mitosis notes. All right, this is just going to reiterate what you just wrote in your notes, so you can put your pen or pencil down and just make sure that you understand this idea. So cell division is where these cells are going to divide into two new cells, okay? They need to divide because living things grow by producing more cells, not because their cells get bigger in size, okay? And number two, you've got to repair that damaged tissue. If you look at the diagram to the right, you'll see a picture drawn of someone who has a cut. Okay, so let's say that you're out riding your bike or your skateboard and you fall and you skin your arm or you cut your arm. That opens a hole in your skin, or what this calls epidermis. Now, if you never repaired that tissue, you would just free bleed or just walk around with a big hole in your arm. But what happens is, like we saw with the blood cells in previous lessons, you get your platelets, they plug up the hole, which keeps you from bleeding to death. Then your skin cells immediately go to work in repairing that damaged tissue, and it forms a scab over that opening or that hole. Underneath the scab, and you guys, this is why you're not supposed to pick your scab. Underneath the scab, new skin cells are being regenerated and woven together. Once that scab falls off, you then have this nice new skin. The third thing is if a cell gets too big, it can't get enough nutrients. It can only work at a certain size. And so if it can't get nutrients in and it can't get waste out of the cell will literally just die. So they have to reproduce themselves before that happens. Pause the video if you need to right here and make sure that you understand the notes that you previously just wrote down. All right. So the next thing you're going to add to your notes is this, mitosis. The definition of mitosis is the division of the nucleus. It's not just, there's something else that happens that divides the cell. So mitosis is specifically the nucleus or part of the cell that divides. This occurs in every body cell. Every body cell. So what does this mitosis accomplish? Well, what it does is it takes one old cell and then it divides to produce two brand new cells. And these two new cells each have a nucleus that have their full set of instructions or chromosomes. And we talked about the other day how important it is that your body always carry your recipe from cell to cell. And that if it doesn't have a good recipe, that's when you have problems. Go ahead and pause the video right now to write these things down in your notes. All right, just going over what you just wrote down in your notes. Mitosis is the division of the nucleus into two, what we call nuclei, each with the same number of chromosomes and the exact same recipe. Mitosis occurs in all of your body cells. So what does mitosis actually accomplish? It makes new, what we call daughter cells. These new daughter cells have a nucleus with a complete set of chromosomes. And I know there's that daughter cell being a new vocabulary word. Just wait, because we're going to get to that. We'll come back to that and you'll write that in your notes. Okay, but just as an aside, you have this parent cell here, and when it divides, it makes two new cells, and we call those cells daughter cells. All right, the original cell, like I just said, is called the parent cell. The two new cells are called daughter cells. So in this diagram right here, you've got the parent cell at the bottom. It copies its DNA. So now you see it's got two different copies of that. Let me get my little highlighter so that you can see. So here it's got two copies in this one nucleus. Then the nucleus begins to divide in mitosis, and it takes a copy to each side. Once that nucleus divides in half, you now have two complete copies that are individual or by themselves. And so we call those the two daughter cells. Before cell division can occur, the cell has to do what we call replicate. Replicate, and that means it copies. It makes a copy. All of its DNA is copied and hopefully exactly right so that they get a complete set of genetic information from that parent cell. Each daughter cell is exactly like the parent cell. It is the same kind of cell and the same number of chromosomes as the original cell. So if you have a muscle cell that goes through mitosis, it's going to produce two exact same muscle cells. It's not going to like randomly produce a heart cell. It's not going to randomly produce a blood cell there. It's going to produce the exact same thing. And remember, that's part of the cell theory, that all cells come from pre-existing cells, and those cells will make the exact same kind of cell that they were. Let's go ahead and pause the video here so that you can add this portion to your notes as well. If you're highlighting, I highly recommend you to highlight the word replication right here with its definition. All right, so now we're going to actually start the whole process of mitosis, okay? And that's where we begin with interphase. Interphase is the first phase. Don't write anything down until I tell you to. I'll give you full instructions. Just watch these diagrams. Interphase is the part of the cell, the phase of the cell in which it spends almost its entire life in. It will stay there and it will sit and do its job. So if it's a brain cell, during interphase, the cell is going to be transmitting all of those different pulses and communications. If it's a blood cell, it's going to spend almost its entire life in interphase, delivering and picking up waste, delivering oxygen, etc. Okay, and if you look inside this nucleus, you have just this chromatin that's just a crazy mess. It's not organized. It's obviously not copying anything. It's just kind of sitting there doing its job. So in your notes, let's go ahead and let's add this. You're going to use this whole circle as your cell and draw the nucleus inside of this cell. And in there, it's uncoiled DNA that's just kind of crazy and unorganized. The facts that you want to write down about interphase: 1, this is where the cell grows and develops. It's also going to spend all of its time doing its cell jobs. Almost its entire life is spent in interphase. At the very end of interphase, it's going to replicate or copy its DNA. Go ahead and pause the video here and write this in. All right. So interphase is that growth, is the growth of the cell. It's where it develops. The DNA replication or copying occurs during this time, and it grows and carries out all of its normal cell activities. It also will replicate all of its other organelles as well. So if it has a mitochondria, it's going to make another mitochondria so that it has double the organelles. It'll have 2 vacuoles. It'll have multiple lysosomes, etc. Cell, and this is super important. I've said this several times, but make sure you have this written down in your notes or underlined. It spends almost its entire life cycle in interphase. Okay, so after interphase, it then is going to enter what we call prophase. Now, from this point forward, this entire process takes about 18 hours. So literally, mitosis is the last 18 hours of a cell's life. Okay, and that's an approximate because things can go a little faster, go a little slower. It just varies depending upon the cell and the environment that it's in. Okay, so now it's going to actually start mitosis, and it's going to enter what we call prophase. Okay, in prophase, if you look at my diagram, and you're going to start drawing this in just a minute, all of that DNA has now been copied and is in those chromosome pairs and those little X's. Okay. We also have these things called spindle fibers that develop, and they go from one side of the cell and they connect it to the other side of the cell. And this is going to be really important when it's time for the cell to completely divide in half. These sides of the cell are called centrioles. The chromosomes coil up and make those nice little X pairs. So make sure under the facts you write down centrioles. They stay by the nucleus, and it is involved in the development of those spindle fibers. The nuclear membrane disintegrates, and the chromosome pairs coil up. Go ahead and pause the video right here and get this into your notes. Okay, here's a picture of what you just wrote in your notes. This is showing you just a little clearer. The nuclear membrane is here. This is showing you the outside of the cell. You've got those spindle fibers right here, and then you have those chromosome pairs that are kind of hanging out in this area with the spindle fibers. Just remember that those chromosomes coil. The nuclear membrane begins to disintegrate, so it's going to start getting really thin. Okay, the spindle fibers that are made of microtubules are now formed and ready to do their job. This is just another picture of what we just talked about, prophase. Okay, looking at the centrioles. Minute cylindrical organelle near the nucleus in animal cells. It's occurring in pairs and is involved in the development of those spindle fibers in the cell division. Those spindle fibers are going to be the things that literally snap the cell in half. So this is super important. And those centrioles are the things that make those spindle fibers, and they connect to each other on each side of the cell. All right, so after the cell has finished prophase, which means that right here it coiled up its DNA, and then it began to form those spindle fibers, and the DNA pairs are hanging out in the center. It then enters metaphase. Okay, metaphase. And the thing I want you to remember about meta, it should sound like middle. So metaphase is where those chromosomes are going to line up down the center of the cell, like right down the center. And they are attached to these spindle fibers by these little buttons right here that are called centromeres. Centromeres, okay? Each part of this chromosome X is called a chromatid, okay? So, facts about metaphase. You want to write down that the centromere, it's the place at which the chromosomes connect to the spindle fibers. The chromosomes line up in the middle of the cell, and they are preparing to be split in half. Go ahead and pause the video right here and take all of these notes down and step 3 of mitosis. Okay, this is just another picture that's going to show you all of those things we just talked about. And I really love this photograph of a cell going through mitosis right here, because you can literally see the line of chromosomes lined up down the center of the nucleus in metaphase. Okay, and you're going to be able to see this in your cell lab, your mitosis lab on Wednesday. So if you look right here at this chromosome, you can see the sister chromatids, which is each side, Okay, so you have one side here, you have one side here, and they're bound together at the center like a belly button. Okay, I always think of it as like a belly button, and it's called the centromere. Those centromeres are going to be attached to the spindle fibers, and those spindle fibers are going to be attached to these centrioles, which are going to start pulling. Okay, but for metaphase, they're just aligning down the middle. Here's another picture showing you those centromeres, the belly buttons, the point at which the chromosome is attached to the spindle fiber during cell division. Pause the video if you feel like you need to make adjustments to your notes right now. Okay, just don't forget that this is where the chromosomes line up in the middle of that cell, and that those spindle fibers are connected to those chromosomes at the button part, which is the centromere. All right, here's just another slide that's just reminding you of the centromere. At this point, you should be thinking, wow, this is a pretty important thing because she's said it 3 times. Make sure this is in your notes. All right, so after metaphase is complete, okay, it's been a couple of hours and it's gone, the cell nucleus has gone through metaphase, we then enter the phase called anaphase. Anaphase, and anaphase starts with an A, and so you should think of apart. Anaphase, apart. Okay, this is where those spindle fibers are literally going to be snapped in half by being pulled with those centrioles, and when it snaps in half, it pulls half that recipe on one side and the other part of the recipe or copy of the recipe to the other side. Okay, the chromosome copies divide, and the spindle fibers pull the chromatids to opposite poles. Okay, so now on this side you have a complete recipe here and a complete recipe here. So you can see how it's preparing to be ready to divide the entire cell in half, and they would both have a complete recipe. Go ahead and pause the video here and write this down and draw this in your notes. All right, here's another slide that shows you, and again, I love this picture. This is an actual microscopic photograph that shows you a nucleus going through anaphase, and you can see right here where those spindle fibers have pulled the chromatids to opposite poles, and it's stretching out that nucleus. Okay, here's another drawing, which I think is a really good representation. If you need to make changes to your drawing, now would be a great time to do that. An anaphase is defined as the point where chromatids separate. The number of chromosomes have doubled so that you have a complete recipe on each side. Anaphase meaning apart. Don't forget to have that in your notes, that it means apart, and that those chromosome copies did divide, and the spindle fibers pulled the chromatids to opposite poles. They then are called chromatids, and that's why I made an adjustment to this right here. You can see that because they're no longer chromosomes, because half of one is on either side. They've been separated. All right, so after anaphase, we go to telophase. Telophase, or some people call it telophase. I've heard it both ways. And this is where things are going to ultimately get really close to completely divided. Okay, telophase is, and please use your maturity here, because yes, I know that this word has other meanings, but this is when the cleavage develops in the cell, and that's where the cell begins, the nucleus begins to pinch in. Okay, and that's going to be right here. And so you're going to need to draw that in and draw that in right here and label that as the cleavage. And now you have nucleus or nuclei on each side of the cell or of the nucleus. You have a nuclei over here, a nuclei on this side, and now the entire cell is pinching in, getting ready to divide in half. So the facts that you're going to write down is that those chromosomes uncoil, as you can see right here. It's that crazy mess again. The nuclear membranes now reform, the spindle fibers disappear, and now we have two very separate and independent nuclei. Pause the video right here to write this down. All right, so here's some more pictures. This one shows you really well. Of course, I did color over it, but it showed really well that that nucleus had completely divided and that the cell right here had almost all divided. It's got the cleavage here at these pinched insides. You have your nuclei here, and all that's left is right here for it to snap in half. Okay, and when it does, when the cell does actually snap in half, those are going to be the two daughter cells. Now notice in telophase, they have not snapped in half yet. The cell hasn't. We just simply have two distinct nucleus. Okay, so the last and final thing that's going to happen in this whole process is actually a separate process. It's kind of part of metaphase, but it also stands, or not metaphase, I'm sorry, it's part of mitosis, but it does also stand alone. And this is cytokinesis. Cytokinesis is when the rest of that cell actually snaps in half. It actually divides and lets go of the other one, and they become two independent daughter cells that are no longer connected. Go ahead and pause your notes, or pause the video so you can put these in your notes as well. All right, this is showing you telophase because it hasn't snapped in half yet. It does show you that the chromosomes have uncoiled, and now it's kind of that crazy mess. The nuclear membrane forms here again in telophase. If you want to go back and add that to your notes, you now have two nuclei, and those spindle fibers have disappeared. Then at cytokinesis, they actually completely divide. And once the division of the rest of the cell happens, then the cell immediately goes back into interphase. Both of these cells. So after cytokinesis happens and they've actually separated, the cell goes into interphase and grows, performs its cell activities, and does all of that stuff until the last 18 hours of their lives. And then this whole process will start over again. All right, so this is just a really nice summary drawing, and this is also, I put this drawing in your test review notes, so you don't need to try to copy this down, and you don't need to ask for a photocopy because you're actually going to get a copy of this. Okay, now cell division, they have their normal amount of times that they do this. Okay, if a red blood cell lives for about 4 months, then after that time frame, it's going to go into its 18-hour mitosis. And that's what's normal. However, you can have problems. Remember, I talked about when they copy that recipe, you can have problems that happen. And that's the thing we don't want to happen, but sometimes it will. And if you look at this right here at the top of this, you can see the initiation, which means the first problem. And we call that a mutation. The recipe was copying and it made a mistake. And when it made a mistake on this recipe right here, this cell is going to go through mitosis again. And when it does, it's going to provide a bad recipe, a bad copy. So here it goes again. It copies it and it makes another mutation. And so it keeps mutating and mutating. And as it continues to divide with a bad copy, that's how you get cancer cells. They're cells that continue to go through mitosis with bad copies of a recipe. The DNA controls all cell activities, even its cell division. And so when some cells lose their control, they lose the control over how fast they divide, They start dividing quickly, and their DNA becomes damaged, like I just went through, and then they start dividing with bad copies. And so literally, you guys, this is the definition of a cancer cell. It's rapidly dividing cells that have bad recipes. These super dividing cells will then make a big mass, and these masses are called tumors. Now, not every tumor is going to be cancerous. You can have benign tumors that are not cancerous. And so these cells don't spread to other parts of the body. And you can see a picture of cancer cells rapidly dividing in one spot, and the normal cells all around it. And you can see how that's really, really thick right here. And that is that tumor that's forming of these poor cells. Now, when those cells have cancer, when they're cancerous, we call those malignant tumors. And so these cells can break loose and go and invade other healthy tissues, and that's what we call metastasis. Metastasis. So if you've ever had a relative or have known someone and they say, oh, their cancer has metastasized, that means that these cancer cells down here, some of them have broken away and have gone somewhere else in the body and invaded new tissue. And that's a really upsetting diagnosis to hear, because that means the cancer is spreading. Okay, go ahead and pause the video here and write down all of these new vocabulary words, so that you would write down that benign is not cancerous, and they do not spread to other body parts, that malignant is cancerous, and that these cells can break loose and invade and destroy other healthy tissue, and that process is called metastasis. Okay, this is just showing you some pictures of benign tumors that are usually self-contained. And what they'll go in and do is a lot of times they'll just go in and cut these out right here, and then the body will heal. It's just like it's a little surgery, and the body will then heal this hole that's been put here by taking out those tumor, that benign tumor. Now here's a picture down here of a malignant tumor that has now metastasized right here. You can see that some of it broke off and is now entering a blood vessel, and it is now going to travel. And so a lot of times, especially like with breast cancer, they'll go in and they'll have surgery to remove the lump. You've probably heard that before. Oh, they went in to remove the lump out of her breast. And so they go in, and what they're hoping they can do is cut out all of this cancer and not leave any behind. Because if a little piece is left behind, then it can travel and go elsewhere. Or when they're cutting it, they don't want to make any of those cancer cells loose off of that tumor and then spread again. So that's why those cancer surgeries are so scary and so meticulous. They have to be so careful that they get everything and make sure that it hasn't already traveled somewhere else. All right, so cancer is not just one disease, but it's a bunch of different diseases. There's like over 100 different kinds of cancers. And so this little diagram right here is just showing you a few of them. You can have cancer, you can have lymphomas and leukemias, and you can have sarcomas, which are in your connective cell tissues. You can have cancers located in organs. You can have it located in just actually any kind of cell is a candidate if it has a poor recipe and starts failing to control it, how fast it replicates and how fast it divides. It has the possibility to become a cancerous cell. This is just a grid showing you the prevalence of cancer worldwide. Now, this is an old statistic grid. It was like from 1997, but still you can obviously see right here, breast cancer is one of the most aggressive cancers and one of the most prevalent cancers. It's up here past or right at 8 million cases a year. You can see that the next one would be possibly prostate cancer is pretty high. You can see that cervical cancer is really high. And depending upon the cancer and the treatments determines the death rate. This is not the death rate. This was just cases. Some cancers are more deadly than others. Breast cancer can be very deadly. However, if it's caught early enough, they can get in there and they can have a nice success rate. Whereas with cervical cancer may be, there may be less cases, but it tends to not have a treatment rate as high as a lot of the other ones. It tends to be more fatal. And then you can see all these other different parts of the body as well. All right, so I'll show you this one at some point. This was called The Immortal Life of Henrietta Lacks, and I think that when you did the cell stations, you got to read about how the government actually stole her cells. But it did end up being a really positive thing because it helped them understand cervical cancer. So this is a grid that will tell you that in interphase, it just kind of tells you main things that happen. And in each phase, we'll go through that really quickly. And just as a review, interphase is where the DNA copies itself. It replicates and it grows and replicates its organelles. Then it hits prophase. The chromosomes coil up. Then the nuclear envelope or the nuclear membrane begins to thin and then disappear, and you get those spindle fibers. Metaphase is where those chromosomes line up in the middle. Remember, they line up down the center, and they're attached by those little centromeres, the little belly buttons. And they're just kind of getting all organized and ready for that nucleus to divide. Then in anaphase, remember, ana or A is apart. Those chromosome copies divide and they're pulled apart by those spindle fibers so that you get chromatids on one side and chromatids on the other. They go to opposite poles. In telophase, the chromosomes uncoil. They change back into that kind of crazy looking chromatin stuff. And the nuclear envelope or the nuclear membrane reforms, and now you have two independent nuclei. And then cytokinesis happens, and this is where the rest of the cell divides. I'm not sure why that says chromatin. Let's just ignore that right there. The rest of the cell divides. It completely pinches in, and so now you have two independent cells that are ready to do their jobs.